All right, welcome everyone. Thank you all for joining us. We're so excited to have everyone here for this rare opportunity to hear from a researcher. Just over 3 years ago, Colby was diagnosed with lymphoma, and after nearly 800 days of chemo, just this weekend, we celebrated 1 year off treatment. Yay. Colby's experience inspired us to do our part to try to change the face of pediatric oncology. Early in Colby's journey, we were lucky enough to meet the amazing folks at the Pablove Foundation and decided to work with them to fund our own research grant in Colby's honor. We kicked off this grant in the summer of 2021 and fully funded the $50,000 in seed grant in just under 9 months. We were thrilled to be able to work with the Pablove Scientific Advisory Board to select the best science. Just 1 year after completing our funding, not only have we selected and awarded this grant, but this funding is already producing some promising results. Before we introduce Dr. Bahrampour to talk about these amazing results, I'd like to turn it over to Colby to share a few words.
Thank you for joining us. We are so happy to be hosting this event, and we really hope that you learn a lot tonight. When we were first deciding what type of grant to fund, we didn't want to just pick anything, or when we were deciding what type of grant to fund, we didn't want to just pick anything or to focus only on what type of cancer I had. The truth is, I'm one of the lucky ones. Despite everything I went through, there are so many kids with much more aggressive cancers that don't have many treatment options. The grant we picked has a real promise to change the landscape of pediatric brain cancer. You may not know, but May is actually Brain Cancer Awareness Month. So without further ado, I'm going to pass it back to my mom, who will introduce our very special guest.
So it's my honor to introduce Dr. Shahrzad Bahrampour, who is a postdoctoral research fellow at Sick Kids Hospital in Toronto, Canada. She completed her undergraduate training in Iran and received a PhD in Sweden. She currently works in the lab of Dr. Wong, the Canada Research Chair in Cancer Biophysics. Her vision is to make a significant contribution to the field of brain cancer research by identifying novel therapeutic approaches. She has spent the past 14 years studying neurodevelopment, stem cells, and cancer in her efforts to turn that vision into reality. Now we are lucky enough to hear more about one of these promising and innovative treatment approaches. Dr. Bahrampour, thank you for being here, and now please take it away.
Oh, thank you so much for such lovely introduction, and I'm so excited for being here today. And I cannot thank you enough for basically making this possible. So what I'm going to talk about would have not been possible without the support from Pablove, Colby, and Courtney, and the superheroes here, right? So I'm talking about my project. If you can see my screen, let's make sure that actually you see my screen.
Yep, got it.
Amazing. So I'm researching on basically finding new therapeutic targets to help kids that are fighting and in battle with brain tumor, as you mentioned. And before getting to business, let me know how you feel today. So I'm very excited. I'm kind of number 2 in a scale of babies. And in the next 20 minutes, we're going to talk about the importance of developing innovative and effective, more safer treatments for kids in battle with brain tumor. And that's why we are here today. And I'm going to talk about medulloblastoma that my research focused on. And I'll tell you a major challenge in brain tumor treatment is blood-brain barrier. And I'll tell you my findings, and what we found here seem to be a breakthrough in crossing this barrier, and we hope to find this treatment in clinical application in the near future. So brain tumors are unfortunately the deadliest form of childhood cancer, and although they are not the highest diagnosed form of cancer in kids, yet they have the highest mortality rate. And it's because in the past 3 decades, we could not really enhance brain cancer therapy, and therefore we could not reduce the mortality rate. There are many different types of brain cancer, childhood brain cancer, and my focus is on medulloblastoma. being the most common malignant pediatric brain tumor. So today, a standard care treatment for this disease is started by surgical resection, followed by radiation and chemotherapy. And I want to tell you that the problem is anti-cancer drugs are administered at such a high dosage that result in drug toxicity, yet it has very limited therapeutic benefits. And we have to find better, safer, more effective treatments for kids that are in battle with brain tumor. So unfortunately, more than 30% of the kids would lose this battle to medulloblastoma. And the one who survived this, they would have lifelong side effects, which are not even from the disease, but they are from the harsh treatment that kids with medulloblastoma has to take if they are lucky enough to survive. And I don't want to give much detail about that, but I really encourage everybody to hear this unmissable TED Talk from Courtney. I mean, I can't put it in a better way, but she really has raised awareness on how much we have to find and how much urge we have to find better, safer therapeutics for pediatric cancer, in my case, brain tumors. And to find, now that we know how important it is to find more effective treatment, to find a solution, we need to know the problem. And the problem for the major obstacle for treating brain tumor is called blood-brain barrier, or I'm going to call it BBB in the rest of my talk. So to tell you what is BBB, I will give you a metaphor. Imagine the vasculature system in our body as highways and roads. So in our city, in our country, we use highways and roads to connect, to transport, to send drugs and foods from a place to another place. We cannot imagine that we open the door of our house and it gets to nowhere, right? So for a functional community and a city and a given country, we need to have functional roads and highways, but they are not free everywhere. So we have regional regulation of highways, for instance, border control, which is necessary to keep our society secure and safe from foreigners who are entitled to enter. So it has a selective permeability and it regulates the traffic. So we need that for a functional country. The same would be as our vasculature system throughout our body. So imagine that vessels are running around into our whole body, and everywhere is free road apart from the brain, because brain is the master of the body, and imagine that as the border control happens there. So here in this schematic, you would see a side view of the brain, and because it's a very metabolic organ, it needs oxygen and nutrition a lot. So vessels are branched into every corner of the brain tissue. And in a healthy state, if we look at this vessel passing out in the brain, if we look in closer look in zoom, we see this is the blood vessel, and these are the wall of the vessel. The blood cells and molecules such as oxygen and nutrition are passing through the blood, and it's surrounded by brain tissue. But sometimes we have toxic molecules and blood-borne pathogens that are passing through the blood, and we don't want them to get to the master of the body, in the brain. So that's where blood-brain barrier occurs. So basically, it's a defense mechanism that is crucial for a healthy, functional brain, and that protects our body, the master of the body, from entrance of toxic pathogens and toxic molecules. So we need that. But that is an obstacle when it comes to brain with tumor. So imagine here, a tumor is initiated and is progressing, and blood vessels would pass through the tumor mass as well, the same as they go through tissue, brain tissue. So when we want to treat a case of brain tumor, drugs are administered through the blood, circulate through the body, and it gets to the region of interest that we want the drug to have an effect. And the problem is now blood-brain barrier consider the drug as foreigners. They would inhibit entrance of anti-cancer drug from the blood to the tumor tissue. And that is why we have such a high side effects and drug toxicity when we have anti-cancer treatment for brain tumor patients, yet very limited therapeutic benefits. And that shows we need to find novel and new treatment for brain tumor. So I want to give you a brief history. How do we know about blood-brain barrier? It goes back to more than a century ago, and we know about blood-brain barrier based on Paul Ehrlich's findings. He's been a physician, and in his research, he used different dyes, inject them through the blood of guinea pigs, and then he looked through different tissues and organs. So here is a guinea pig embryo. This is actual image from Paul's studies. It's an original image. So this is the leg and the hand. This is the digestive system of this guinea pig. And you can see the dye in the blue that he has injected got penetrated into entire body apart from brain. Brain stays free from the dye. And he noticed that with majority of the dyes that he injects through the blood, brain is the only part that stays free from the dye. And he thought it's because brain doesn't absorb it. But Goodman, in 1909, did a smart and simple experiment. He used the same dye, but directly injected it into the brain. And what he sees here is the slice of the guinea pig brain. And he sees that this is also an original work from Goodman. And he sees that the brain can absorb the dye if it is directly injected. So he found that there must be a barrier between the blood and the brain tissue. And based on this study, we know that, okay, blood-brain barrier happens on the blood vessels that passes through the brain and nowhere else in the body. And I really want you to appreciate how much brain here is free from the dye, and the penetration of the dye is in entire body of this guinea pig. The same happens with the drugs. When we want to use anti-cancer drugs to target brain tumor that sits in the brain, drugs are administered through the blood. It gets everywhere in the body, but doesn't reach the place that we want to target. And that's the problem, right? We have to break this barrier if we want to have better, more effective treatment for kids battling with brain tumor. And BBB hinders more than 90% of anti-cancer drugs. That means basically all anti-cancer drugs are inhibited by BBB. So this is such an urge to find targetable molecules to increase BBB permeability, therefore enhance brain tumor therapies. And I'm shifting gear here. So I want to bring your attention on the fact that about 20% of US FDA-approved drug that we can get from drugstore and can be prescribed today are targeting an ion channel. So what's an ion channel? Ion channel is a big family of molecules that is present throughout our body. They are many different types and involved with many different biological events and processes. And I want to tell you, these are important family of molecules, as much as 2021 Nobel Prize in medicine was given to Paraputian for his work on one ion channel called piezo-1. So that's interesting. But why am I saying this? It's because sad news, there is no anti-cancer drug that is FDA approved that target an ion channel. So it's a big missing. And in my research, I focused on finding a targetable ion channel that we can use and break this BBB barrier to help that enhance the efficacy and efficiency of brain tumor treatment. So the hypothesis of my work is basically, I want to target the ion channel, a chloride ion channel, to enhance BBB permeability, give better access to the drugs from the blood to the tumor site. Therefore, we would have more effective and safer brain tumor treatment. So to test my hypothesis, I'm using genetically engineered mouse model, or GEM, for medulloblastoma. So medulloblastoma is a cerebellar tumor. And what does that mean? Here is a side view of a human brain. And then in this back part, you can see this is called cerebellum. It's a crucial part for our body. It regulates the movement and balance. And here is a mouse brain, again, side view, and this is the cerebellum. So in the cerebellum is basically where medulloblastoma initiates. And I'm using this genetically engineered mouse that we have manipulated their genome in a way that when they are in their embryogenesis and then like their fetal stages, when the development of cerebellum should happen. In this case, instead of developing a normal cerebellum with this stereotype anatomy, they actually develop medulloblastoma. So these mice are born with medulloblastoma, and we are using that to study this specific chloride channel and changes on BBB permeability. So how do I even actually study BBB permeability? And in brackets, I want to add, in my talk, I will use this word knockout, by which I mean loss of function. So when I say chloride channel knockout, I mean that we don't have the function of this chloride channel in the tumor. So I use this GEM model and I looked at BBB permeability. How would we actually measure BBB permeability? We're doing something similar to what Paul Rich did, basically injecting a dye, which we know that it's impermeable through BBB. Here I use cadaverine. You don't need to know the name. Just know that it's a dye that is not permeated by the BBB from the blood to the brain. And magenta shows blood vessels, and the green dots show the presence of the cadaverine dye into the normal brain tumor. So now I compare that by a mouse tumor which has knockout of this chloride channel, and this is what we see. This was striking. Now you see when we target this channel in the tumor, it enhanced the access of cadaverine dye from the blood to the tumor site. So that shows that BBB is broken, the permeability is increased. And I didn't leave it at that stage. I was wondering that if this barrier is broken enough to allow bigger molecules also enter from the blood to the brain tumor tissue. I checked for another molecule called even blue. The same as expected in normal tumor, we have very few dots green here that shows presence of the dye in the tissue. And then by targeting this channel, we give better permeability and access for the dye from the blood to the brain tumor. And the last, I checked for fibrinogen, which is much, much bigger molecule, by which I obtained the same result. And this data shows us that we could break successfully the blood-brain barrier and give access to a high range of molecular size. That means that we could break this blood-brain barrier. And that means that we can give access to a vast range of anti-cancer drug with many different molecular size from the blood to the tumor site that we want to have this therapeutic effect. So this was a eureka moment for us. And then I went to the human patient data and I wanted to see if there is any correlation between loss of function of this channel and survivability of the MB patients. So what I noticed that the blue line here shows that patients, MB human patients who has low function of this chloride channel respond better to treatments, therefore they survived better. So this supports our hypothesis that lower function of this channel, higher BBB permeability, better drug access, better therapy response. And I wanted to test that in my GEM model. So I went back to this mouse model that I just explained, and I used vesmodegib, which is a small drug that is used to basically treat MB. And then I designed my experiments into two groups, normal tumor and targeted tumor mice. Each group was divided to two subgroups, the one that received no treatment and the one which was treated with vesmodegib. As we expected, when we treated our mice with vesmodegib, in the case of normal tumor mice, there was no benefit, there was no therapeutic benefit for treating the mice with vesmodegib because it's not permeable through BBB. So basically the drug cannot get to the tumor. So the mice died the same time as the no treatment mice. But when we target this chloride channel, the targeted tumor mice treated with vesmodegib lived twice longer compared to the control group with no treatment. And this is huge, like twofold changes. Here in case of GEM model, we are talking about days. If we translate this scientific data to human, that would mean years. And this brings hope that if we can use this as treatment for our kids, that would grant them so many more birthdays to celebrate. And this was a hallelujah moment for us. And not only that, but also it would give much more happier lives, because vismodegib has side effects as any other anti-cancer drug. And the known side effect for vismodegib is bone toxicity. So MB patients are kids that their skeleton is not fully grown, and they have a long to live, and they have to grow. And then this treatment, the side effect of vismodegib, would slow or stop children grow by affecting their bones. But we propose that using and targeting this chloride channel would make this medicine, vismodegib, much more effective at lower dosages that would basically not only have increased the number of survival, but also it would grant them with a happier life with very few or even no side effects from the MB treatment. So now that we found this solution, we could break BBB barrier, this old problem for treating brain tumor. We wonder what is the mechanism of action. And for that, as I mentioned, this chloride channel is knuckled in tumor cells. Here in green, you can see the tumor cells are marked, and magenta is showing my blood vessels. So in the top panel, it's a normal tumor. These are the sections of the tumor. And then comparing the normal tumor cells are close contact to blood vessels, but they are not really interacting with them. When I target the chloride channel, that was to our surprise. We find that tumor cells are coming in close contact with blood vessels as if they're attacking them. And I stained for other markers. Again, green is showing the tumor cells, magenta is the blood vessels. And I found that at the site where tumor cells are in contact with blood vessels is the site that we have seen the change in BBB permeability. So this is very interesting. We find that we can use the enemy, being the tumor cells, to actually open the door and break this obstacle, give access to the anti-cancer drugs from the blood to the tumor site. So the enemy comes to help here, and that was very interesting for us. And I went one step further and used electron microscopy to look at this histology at much more higher magnification. This is nanometer. And in the top panel, this is basically a cut of blood vessel. This lumen is where the blood passes. And in the yellow, you can see the cells that basically builds the blood vessel wall. So this is a very stereotyped case of a blood vessel in the brain. And this is example 1, example 2. This is a more zoom in close for the gating or tight junction, where basically this regulation between the exchange of the molecule between the blood and the brain happens. So that's very stereotyped in our normal tumor. And when we target this chloride channel, we find extreme and dramatic changes in the endothelial morphology. They lose their shape and apical and basal polarity. The lumen is malformed. And when we look closer at these gates or tight junction, we call it, we see that there are a lot of openings and breakage, and that's where drugs and anti-cancer molecules can get from the blood to the tumor side. So it's okay if you are not familiar with the morphology of basically capillaries and blood vessel, but I want you to just appreciate the dramatic changes in the morphology. So this is not functional as it should be before, and meaning that we use brain tumor cells to come in contact with the blood vessels and break this barrier. Now the gatekeepers are permission givers, and basically this barrier is broken. So to summarize, in case of brain with tumor, we have the blood vessels that are passing through the tumor mass, and then we treat the cases basically systematically with anti-cancer drug through the blood, and then drugs are passing inside the vessels. we propose by targeting this specific polarized channel, we would permit an access the drugs from the blood to the tumor tissue, and that would help to have more patients survive and fewer long-term side effects of the treatment. So just to give you an overview of how any therapeutic target is found and is now available in the drugstore and easy to be prescribed, it's actually many years behind in the basic labs, and that is where my research stands. So we did a lot of years in the lab, spent a lot of years to identify this target, and I did in vitro and vivo studies to find out the effects and mechanism of action, and we are hoping to get in future in clinical trials and bring this to our patient bedside. And one specific point that is a positive point about my research is that we already have 3 FDA-approved specific inhibitors for this polarized channel. And this is outstanding because many basic research has been done on BBB, and they identified molecules that can manipulate BBB permeability, but we cannot use that in clinical applications because we don't have a specific inhibitors for those molecules. And in this case, we already have 3 FDA-approved specific inhibitors that brings us more hope that we're going to have in near future using this therapy to help kids that are in battle with MB and even other brain tumor types. So this work and work like that, which are studying BBB, would not only help cases with Medulloblastoma, but other types of brain cancer would also benefit, and hopefully even other neurological disorders, because that's an obstacle for whatever is the issue and problem inside the brain. We need to give access to the drugs from the blood to the region of interest. And to summarize, we find the specific polarized channel that we can target and manipulate to enhance and break this BBB obstacle, granting VIP access for the anti-cancer drug from the blood to the tumor site. And our GEM model shows twofold increase in survival, which translating that to human offers hope for many more birthday to basically celebrate for kids that are in battle with MB. And it would also give us hope to grant them with a happier life to live because of no or lower side effect from the treatment. And then having 3 FDA-approved drugs specifically targeting this channel brings us more hope that we can have this research closer to clinics. And at the end, I want to say that the brave kids that are in fight with MB or even other brain tumors and even other neurological disorder, hopefully would benefit from our knowledge, and hopefully we can bring this knowledge into application and into clinics. So I would like to thank everyone in my lab who has been great support throughout these years, and some of them are now in the Zoom. And I would like to have a special thanks to Pablove and Colby's and Courtney's, and who without their support, this work would have been just a dream for me and would have not been possible. And I would like to take any question if there is. Thank you.
This is Kirsten. Hello, everybody. I think that was just an amazing talk, Dr. Bahrampour. You just brought the science down to a level that non-scientists can understand, and the breadth of your presentation is just unbelievable. You really build it from the bottom up to what you actually care about and what you know about, and where new breakthroughs will come from for society, really, not just pediatric cancer kids, but this potential of your research is just breathtaking, and we're just really pleased to have you here and listen to your work. So let's all join in with some silent applause for Dr. Bahrampour, please. All right. Hello, everyone. I am Kirsten Lynham. I am the CEO of the Pablove Foundation. And on behalf of the foundation, our scientific advisory committee and donors, we are so privileged to be able to be one of Dr. Bahrampour's early funders. We look forward to watching what happens with this research project and with your career as well. In addition, I also want to thank very specifically the Horvath family and the Colby Strong donors who are here with us online. You all made this possible, and I would like to invite everybody to share a round of applause with them as well. Very impressive. I'm also pleased to see so many families joining us today. And I know that a lot of the families here are personally affected by medulloblastoma or another form of pediatric brain cancer. Many of you are advocates yourselves, and we thank you for the work you do to champion your kids and to champion the need for safer and better treatments leading to a cure, hopefully as well. But as we all know, there is a glaring void, and we all know there is so much research that still needs to be done. And there's just one thing missing, and that is funding. So to switch the page here a little bit, there is an exciting new development here at the Pablove Foundation. And what Colby and his family did, we now offer as a direct pathway for families to feel empowered and to take action. Action to really move the needle toward a cure. Action like what Dr. Bahrampour is showing us here today. I mean, look at the Horvath family. They did all this and they reached their goal in just shy of 9 months with the help of their community. It's super impressive, but on the other side, it's also a sign that it is possible. As a foundation, it is part of our mission, and frankly, it's a part of our hearts here as well, to seek out and fund the most promising, daring, and innovative research and cancer researchers like Dr. Bahrampour. Dr. Bahrampour's project is just one of more than 50 projects we have funded over time, and I believe that our scientific advisory committee is doing an outstanding job identifying the most meritorious research ideas. You can become part of the stellar grant mechanism, and you can partner with us to fundraise and award your grant as well. If this speaks to you, please stay on the line after the show and we'll talk more about that. Now, I'm sure many of you have questions, and I would like to open the floor for Q&A. And to do that, please raise your hand. I'll try to find you on the screen and unmute and ask your question, or otherwise, if you'd rather, you can also write your question into the chat and I will read it. And I'm sure Dr. Bahrampour will just bedazzle us with her smart answers.
Thank you so much, Christine.
So let me see if I can find somebody who has a question. Yep, there you are, Jean. I see your hand up.
No,
Yeah,
that was
I'm
great.
I'm with
I'm
my son.
sorry.
Hello. So we have a question. Aryan is in high school. He's a junior, going to senior. He is very much interested to pursue medicine. He is a survivor of osteosarcoma. In 2018, he got surgery and chemotherapy. So for his career, he is interested in that, in the field. What he has shown today, like a very exciting, he was telling. He asked me, he told me to ask a question. If the brain BB, the brain blood barrier, if it is broken to transmit the medicine, so if that happens, it may carry other toxins to the brain in the blood, so that may affect. So how are you going to remediate that?
First, thank you so much for taking the time to attend this talk, and smart question. So the answer is that absolutely that's a concern for any anti-cancer drug, and when it comes to brain, again, because that's the master of the body, we want to protect the rest of the brain, the healthy tissue, right? So in my case and in my research, I showed you that the brain tumors are the gate openers. So basically, we will have the effect of opening BBB on the side of tumor and not the healthy part of the brain. That's one thing. The other thing is that we're going to use inhibitors that are drugs, and drugs have limited lifetime. So we are not doing like gene therapy that would down-regulate or basically knock out the gene that express the channel. We're going to use the drugs that have limited lifetime. So basically, it's as if we have a switch in our hand. We're going to open this BBB for the time that we are administering the drug. And after that, when we have the drug access to the brain tumor and it can have the effect, then we close this valve and now the BBB can go back to as it is. And with this research is specific because the result is tumor site specific, the healthy part of the brain will stay unaffected. If I could answer your question.
Yeah. And how, so you need to, once in the chemotherapy administered in the regular treatment, after administering in 24 hours, they would like to, dilute and take that effect before it affects, toxic to the other organs, right? So are there any mechanism for after administered? Is how long that going to work? And is there anything you would like to remove that or just leave it there?
That's a good point you mentioned. So that is dependent. Different drug has different basically regime. And for this drug and this three FDA approved drug that I just mentioned has never been used for brain tumor treatment. As I told you, there is no anti-cancer targeting ion channel brain tumor or tumor cancer, right? So this has been used for asthma treatment, two of them, and one is anticoagulants, and has never been administered for brain tumor. So basically, we need to do a clinical trial to find out the best regime for combining this therapy with other drugs. And another thing is that this so we are proposing a combinatorial treatment, meaning that we are administering this FDA-approved inhibitor for this chloride channel together with another anti-cancer drug, which in my case, I studied with Medulloblastoma, but we have the plan of also trying other brain tumor specific drugs that are used at the current standard care today in the clinics. And for combining it, first of all, finding the best dosage for this specific FDA-approved inhibitors of the channel, we need to do clinical trials. And the other thing is that we need different regime when we combine it with different anti-cancer drugs. So Medulloblastoma is not a cancer drug, it's a targeted drug. It affects Medulloblastoma specifically. It's a targeted specific molecular pathway. And we can also do cisplatin, for instance, which is a general anti-cancer drug together with this. So to give a brief of what I said, we need to bring this to clinical trial, and that would let us know the regime of, first of all, this FDA-approved anti-chloride channel, and then that would also differ what is the synchronized with. So whatever anti-cancer drug that is combined with would have different regime of treatment. But in general,
it's very promising.
Yeah, we are hopeful. And in general, because this is supposed to give so better penetration, meaning that we would have lower dosage of anti-cancer, better treatment, right? So that's why we need to find the right regime when we have this synchronized and combinatorial treatment.
Thank you.
Thank you.
Someone texted me a question, and the question is, after your Pablove grant is done, what is the next step for you?
For me per se, or for this project?
For this project, I think is what they meant.
Okay, so that's what we are trying to do. First of all, within the research, still it's work to do. Now at the next step, I'm using still the GEM model that I explained to you, the mouse model. I'm using that to combine with other treatments apart from Medulloblastoma. I'm using also just to, as I just told you, this is supposed to be brain tumor specific. I want to show that by looking at healthy part of the brain and show that it's not affecting the healthy part of the brain. And I'm going to use a xenograft model. Xenograft model is when we have human patient cells digested, and we take basically after a patient is diagnosed, they take biopsy, so we can in the lab have the cells of Medulloblastoma. We grow them in culture dishes, and then we inject them into the brain of a healthy mouse, and that mouse would develop tumor. So these are called xenograft model or a pediatric PDX model. So I'm going to use PDX model, which would be a mouse that carry human envy, and then use these FDA-approved drugs together with desmodegib. And after that would be human trials.
Thank you for that. I think there is a question from Colby as well. Colby, do you want to ask it directly, or do you want me to?
No, Colby's pop had a question.
Oh, he's so cute.
We can read Colby's Pop's question. I thank you for your valuable research. Can modulating the chloride channel for the benefit of cancer drug efficacy have any other side effects in other areas of the body? For example, chloride channel dysfunction and associated with cystic fibrosis. Colby's Pop's a toxicologist too, so we knew we'd ask a good question.
Of course. So I have to say that that is because these are FDA approved and has been used before already. So we expect to see not such a high level of side effect, if any. Yet, again, I mentioned, I think in brief, that I am testing that in my GEM model, looking at not only healthy part of the brain, but also the rest of the mouse. So we're going to do histology on other organs. The mouse that is treated with this FDA approved, we do histology looking at the kidney, liver, other places in our GEM model to just make sure that there is no or low toxicity. Again, depend on the effective dosage that we're going to apply. And another way is that we can basically do direct administration of this drug into the tumor site. So that is another way to apply this, but it would be more challenging. So first we are trying systematic and then looking at the toxicity. So to wrap up what I say, because they are already FDA approved, has been already used for patients to treat asthma, we expect to see very little, if any, side effects. And yet I'm doing more analysis on my GEM model to support this idea.
Thank you. There is a question from Misha, and the question is, what side effects would a CI channel agent have on the patients?
So it's a great question. It's basically, we don't know really, because it has never been targeted. So it's fun to know. I'm very triggered. I'm very inspired by this fact that this channel has been known for 4 decades, and yet we didn't know the essential components of this channel at all. And then until 10 years ago, we found out the gene that is the essential component of this was actually found by this lab that received the Nobel Prize. So far, we know that this channel is expressed throughout our body. And the thing is, it is required through embryogenesis, but we're gonna target this and silence it after it passes the embryonic stages. So we would expect at low dosage and limited time to see either low or no side effect by targeting this channel. But what does this channel specifically do? Is a swell regulated channel. What does that mean? It means that we have our body, basically you have heard that 70% of our body is liquid and it's water. So we have to have a control and a regulation for our cells to keep their desired size. If cells have any changes in the osmolarity of the liquid that is surrounded by and cannot maintain its basically size, would die. So the chloride channel, this chloride channel specific, is a swell regulated, by which it means that it's important for maintaining cell size regulation. And it is mainly important for cells that go through division. And why is that? A cell that want to divide, meaning that it can kind of enlarge and then become two cells, and again enlarge and become two cells. So for that enlargement part, it needs to be able to maintain the desired size again after division. So I expect if there is any side effect, it would be on the cells that are more dividing. And therefore, that's why, again, I think in the normal brain after development, where we have very limited division, we should not have much or any side effect in a healthy brain. And also in the entire body, we expect, because there are some redundant molecules that can cover up for this channel for a short period, again, we expect to see low or no side effect. But yet, to give you the definitive answer, we can't know until we do clinical trials and see what we see. But this is what I have, this is my expectations.
Thank you. There's a question from Mighty Quinn. And there's a caveat up front, maybe this is a repeat, but how do you close the BBB for the possibility of future tumors and their ability to travel outside the brain?
Okay, so I think what he or she means that we have brain tumor metastasis that happens through the blood vessels, if that is what he or she is asking for. If you would confirm that I got your question correct, then I can continue. Yes, okay. So if you are wondering that opening BBB would actually increase the case of metastasis, I would consider that not be the case, because we are definitely combining this with the drug treatment at the same time. So, and again, as I mentioned, this opening is temporary. It will get closed after the lifetime of the drug is passed. And then meanwhile that we have the drug treatment for anti-cancer, even if cells can escape to the bloodstream, they would get actually even more access or exposure to the drugs. So we don't expect to increase risk of metastasis, not if would decrease, if anything, then increase because of increased permeability to the anti-cancer drugs.
Thank you. We have time for maybe one or two more questions. There is one that I see here. Dr. Bahrampour, thank you for the interesting and understandable overview of your work. Can you give a few examples of drugs that might be most likely given together with your CI channel agent?
So the first one that we are going to try is actually Vesmodegib. And so I didn't give you this overview, but basically, not only medulloblastoma is one type of a lot of brain childhood tumors, but even medulloblastoma has different subtypes, and it's divided to 4 subgroups. And one is called sonic hedgehog, which my gem model is sonic hedgehog. So there is difference between BTB permeability or blood-brain barrier. In case of tumor, we call it blood-tumor barrier. So blood-tumor barrier in different subgroups of medulloblastoma is different as well. Sonic hedgehog, the group that I'm studying, is the most intact. So that means that they have the highest, basically, permeability, the highest barrier in the case of sonic hedgehog. And Vesmodegib is actually a drug that targets sonic hedgehog specifically, and that's why I said it's not an anti-cancer, it's a targeted molecule, it's a targeted drug. So the first thing that we want to try is actually combining it with Vesmodegib. After that, I'm thinking of cisplatin. And cisplatin is a general anti-cancer drug. It's basically targeting, it's not targeting medulloblastoma specifically, and that is one cancer drug that is having quite a lot of side effects. So we don't want to have high dosage of cisplatin inside the body circulating and getting to other organs. And that's why I want to use that in combination with this FDA-approved drug to see if we can have lower dosage, more efficient.
Thank you. So there's immediate response. That makes a lot of sense. Thank you, from the person who asked the question.
Thank you.
There's a follow-up question from Mighty Quinn. Additionally, what would occur with metastatic tumors if they're in different livations?
What do you mean? What would occur with metastatic tumor? You mean metastatic brain tumors?
I'm assuming that's what's meant. Yeah.
Yeah. So that's I think what you are asking is that if the tumor is at a higher grades and it got to a metastatic level.
Oh, locations. My DeQuinn says locations. So but I think you're answering the right question anyway.
Got you. Yeah. So basically that is the thing. And that is why we need to find, so let me rephrase myself. So why do we have this current standard care division that is surgery followed by chemotherapy and irradiation? It's basically because we cannot use surgery to remove the entire tumor because it is infiltrating, right? And for that, we need to find targetable molecules. So in this case, we are not going with the specula and surgery, it's targetable molecules. So wherever the tumor cells are in close contact with blood vessels, which is entire brain. So no matter how far they infiltrate, no matter how far they metastasize, there will be in contact with blood vessels. And that is the part that we use the enemy. The tumor cells are the one that are opening the gate. So that doesn't matter if we have a nodular or we have infiltrative. Having the tumors close approximately to capillaries would open this barrier, and that would increase the access of the drug. So it would be beneficial for even higher level and higher grades of tumor with infiltrative cases.
Dr. Bahrampour, you made a point that this research that you're doing could open pathways to treat other diseases as well that are difficult to treat, are treatment resistant because of the brain-blood barrier. So this next question, I'm going to choose that as the last question. It blows that up even more. And Misha has the question, if there are opportunities that are opened up by your research for spinal tumors as well.
So good point. That the cells that, again, I didn't mention, so that was a limited time. So I will tell you this. So not only we find that these are tumor cells that are attacking, we have tumor heterogeneity. And then within the tumor, that's why I showed you panels of different markers for tumor cells. We find what cell types in tumor is actually attacking and having this ectopic contact. And these are a type of tumor cells that are called DCX positive cells. So my expectation, and these are not only in Medulloblastoma, we have seen DCX positive cells in other brain tumor cases as well. These cells are, if we have, in the case of that, let's say glioma or like any other brain tumor, if we have DCX in the population, we might be able to trigger the same downstream molecular pathway by targeting its chloride channel. So that's why I say that this can shed light on other, not only brain tumor, even neurological diseases, because these CX cells are one of the most dominant healthy and tumor brain cells. And we can use this mechanism underlying down this chloride channel to basically open up BBB.
It sounds very much like potential. Yes, but we don't know yet. Way more research is required.
Absolutely, definitely. So, and that's why, again, like we are here, right? We need to give more focus on these researchers who do. Yeah, definitely.
Yeah. Way more research is needed across a broad area. And that's why we're here. We here at Pablove and here on this Zoom call, we all have an affinity for research. And I want to thank everybody to spend an hour of your time. Time is a precious resource. Thank you for spending your time with us. Thank you again to the Horvath family for funding this research. Thank you to your donors who are online. And thank you everybody for your interest. And last but not least, Dr. Bahrampour, I can't believe how strong your answers were as well. And all these questions are wonderful. It's an indicator that all these families with kids with cancer, they all get an involuntary science education excellent question. So hats off to all of you, and good thoughts, good wishes, and prayers with all of you. Let's hope that more cures are found, more better treatments are found, so that the community of kids with cancer continues to shrink over time. That's why we are here. We want hope. We want our kids to love and live and celebrate many, many birthdays with us. So thank you very much. If you're interested in the program I mentioned, Fund Your Own Research, please stay on the line and we'll chat some more. And otherwise, I wish you all a beautiful night and a great week. And we will see each other again when we join our community. Good night.